You are now muted. This conference is now being recorded.
Welcome to the AMDA webinar, Design and Performance of Clinical Trials, an Overview. Your speaker for today's program is Dr. Ann Zajicek, Chief of the Obstetric and Pediatric Pharmacology and Therapeutics Branch at the Eunice Kennedy Shriver National Institute of Child Health and Human Development. Operator assistance is available at any time during this conference by pressing £0. A question and answer session will follow the presentation. You may send chat questions at any time using the chat window located to the left of the presentation screen. I will now turn the call over to Dr. Zajicek. Please begin.
Good afternoon. Thank you very much for inviting me to come and talk about clinical trials. We have a lot of pediatric and obstetric trials going on in my branch, so I know a little bit about this area. I will be pleased to take questions while I'm talking. You can interrupt me or just send something in a chat, or you can ask afterwards, or alternatively, my last slide has my name and my email address and my phone number, so you are more than welcome to call me or email me, whatever you want to do if you have some further questions. So the title of my talk is Design and Performance of Clinical Trials, an Overview. I wanted to start by this really interesting article in the New York Times last summer, and they were talking about clinical trials and the purpose of negative clinical trials, because there tends to be a publication bias about negative trials versus positive ones. And the quote was, when you do any kind of trial, you're really trying to answer a question about truth in the universe, meaning that there is a discrepancy between the sample of the people that you're studying and the population, says Hal Barron, the Chief Medical Officer and Head of Global Development at Roche and Genentech. And of course, we can't know that. So we try to design an experiment on a subpopulation of the world, the sample, not the population, that we think is generalizable to the overall universe, that is, to the patients who will use the drug. And I thought that was a really nice way to look at clinical trials, that you're trying to set up an experiment in order to find out the truth of whether something will work on an entire population of people with a disease, even though you can't study everyone with the disease. The model of clinical trials networks tends to be the Children's Oncology Group, which is a grant that goes from the National Cancer Institute, which is one of the institutes at NIH, to the Children's Oncology Group. And this was published in 2012, showing the overall survival of children enrolled. And again, these are fairly recent now, and you compare this to the 1950s and before that, the overall survival for pediatric oncology patients was typically 10% or less. But here we're looking at the differences between 1990 to 94 in the teal line, the yellowish green line, improved overall survival, and 2000 to 2005 even better than that. So what that's shown is that because all children in the U.S. are, or nearly all, enrolled in Children's Oncology Group trials, the purpose of a trial is actually perfectly clear here, that it does in fact improve survival and patient outcome. So I wanted to talk about the type of trials, and I borrowed this from the Wikipedia, which had very good explanations. So we have prevention trials, screening trials, diagnostic trials, quality of life trials, compassionate use trials, but that fourth bullet about treatment trials tends to be the kind of clinical trial that people think about when they're thinking about treating various diseases or therapeutic areas. Now, the subtext about the treatment trial is that you need to understand that there needs to be a background of an observational study or a natural history to study, so that when you have a treatment, you know that the treatment is actually better than the patient who wasn't treated. So that's sort of a subtext of that. But what I'm mostly talking about here is treatment trials. There are several phases, and the first one that I didn't list here is the preclinical study. And the preclinical means that the studies that were done on animals before the first-in-man treatment of the medication. And there is an estimate or an inference that there can be some extrapolation between what was found in the animal model and what will be seen in humans. And most of the time that's true, but sometimes it's not. So the phase, quote-unquote, phase 0, which is sort of a newish term, is a phase where there's microdosing. So in other words, the dose of the drug that's used for the patient or the subject is a very tiny dose. And this is just to get an idea of the time course of the drug concentration after administration, but it's not expected to have any clinical effect. Phase 1 is a dose-ranging safety study, typically in normal volunteers. It depends on how toxic the medication is. If it's expected to be toxic, like a standard chemotherapy agent, then it's likely that this will be done in patients with disease and not in normal volunteers. Phase 2 is a preliminary efficacy and safety study, this time in patients. So this is with patients with the disease. Phase 3 is a pivotal clinical trial to prove safety and efficacy to the FDA, and then Phase 4 is a post-marketing safety surveillance. So rather than going from the sample of patients in Phase 2 and 3, now we're going into the population. So now we're expanding post-marketing safety surveillance. And frequently, well, not frequently, but occasionally there are some surprises here. Given that it takes a fairly large number of subjects to see any kind of safety signal, because again, the reason a drug is approved is because there's a good safety or risk-benefit profile. Sometimes there are unpleasant surprises in the phase or post-marketing safety surveillance. Let's see, the next slide is about clinical trial designs, and I just wanted to point out some terms here. The first one is a randomized study. So the treatment arm is determined by chance. It can be A-B, where the first patient gets treatment A and the second patient gets treatment B, and then the third patient gets A and so on, or A, B, and so on and so on. So those are predetermined by chance. There can be block randomization where there's randomization for the treatment, which is stratified by age or by the clinical trial site and so on. Typically, the clinical trial is called controlled because there is adherence to the protocol, a recording of all safety and efficacy data. If the study is called single-blind, that means that the patient is blinded. Double-blind means that the investigator and the patient are blinded. And placebo-controlled means that the comparator arm is a placebo. The comparator arm can be either an active comparator arm, which would be bullet 1, where an approved product is available for the same indication, or the patient can be treated by standard of care. A treatment arm can also be a placebo, which looks, smells, tastes the same as the active drug but contains no active ingredients. The placebo arm does not mean that patients receive no treatment. It can be the placebo plus the active comparator versus the new agent plus the active comparator. Placebo is sometimes used when there are no acceptable treatments. And it's important to include a placebo in order to avoid bias, especially for behavioral mental health conditions with a high placebo effect where it's difficult to find an objective measure of treatment. Steady endpoints can be a biomarker such as dystrophin in Duchenne muscular dystrophy, or alpha-glucosidase in Pompe disease. Creatinine kinase could be another biomarker. A surrogate marker which has been validated to basically be used in exchange for long-term outcome could be blood pressure or cholesterol for stroke, for example. For endpoint validation, the endpoint validation needs to be clinically relevant. It could be death, event-free survival. Overall survival is usually taken by the FDA as opposed to event-free survival for cancer treatments. For Pompe disease, the 6-minute walk test has been used as an endpoint. However, this is not very useful if you're speaking of a young child or an infant who has not learned to walk yet. These endpoints are generally clinically accepted, and there is a validation process for them. Now, I wanted to talk about one project, which I guess is sort of the seminal clinical trial that showed the necessity of having a placebo. And this was the infamous CAST trial. This was published in the New England Journal of Medicine in 1989, and it was a preliminary report, effective enconide and flecainide on mortality in a randomized trial of arrhythmia suppression after myocardial infarction. Now, the thesis of this project was that if you suppressed arrhythmias in patients who had had a myocardial infarction, that would directly correlate with long-term survival, and that suppression of these arrhythmias would lead to increased survival. The biomarker in this study, again, was the use of the Holter recording, and the biomarker was assumed to be completely correlated with long-term outcome. Now, again, the subtext of this project, and they did throw in one placebo arm, was that the drugs would, of course, be better than the placebo arm. And they did not include an interim analysis here, but what they found out was when they looked at the end results, that following 10 months of follow-up, the patients treated with active drug actually had a higher rate of death from arrhythmia than patients assigned to placebo. On this next slide shows this graph showing survival of the placebo arm actually being very statistically significant, better than the enconide or the flecainide. And again, this does show the point that although the assumption is that suppression of arrhythmia or the active comparator would of course be better than the placebo, and sometimes that is not the case. Let me stop here and answer a question. Hold on. Let's see. The question is, one of the issues we have with trials in Pompe disease is getting people to participate when there is a placebo and when patients often travel long distances to get a study site. Is it really necessary to use a placebo to get good clinical data when adult Pompe patients, even on current treatment, are not improving over time? That is a perfectly reasonable question. So I guess the assumption here is that this is in, I guess, children as opposed to adults. I guess this would be a very specific question for the review division at FDA to discuss the fact that you've attempted, and this has been my own experience, is that to discuss with the review division at FDA the fact that you have attempted to do something and you have a proven track record of the fact that you simply cannot get patients to enroll because of the placebo, because of the distance and so on, and why would there need to be a placebo arm? And that is a perfectly reasonable question to talk to the FDA review division about, the fact that, based on phase 1 studies, based on animal models, based on all the available published and unpublished literature, there's simply no purpose in having a placebo arm because you already know the natural history. So it's not going to be a big surprise that it's, it wouldn't be any different on placebo. So that is a perfectly reasonable question to ask the review division about. I hope that's a sufficient answer. Again, if you want to talk about any of this afterwards, call me, email me, happy to talk. Okay, so the next slide goes into adaptive clinical trial design. And this is a really interesting area. So there's a nice, very long guidance that the FDA put out in, I think, 2009, 2010, something like that, about adaptive study designs. Typically, when you do a randomized controlled trial, you have already decided, what the outcome measure is going to be, what the primary outcome is, what the safety endpoint is, and so on. You've determined the number of patients that you need to prove it, and that's a standard clinical trial. However, with adaptive clinical trials, includes studies that have a prospectively planned opportunity for modification of one or more specified aspects of the study design and the hypothesis based on analysis of data, usually interim data, from subjects in the study. So in other words, you have an interim look, let's say a quarter of the way through the trial. You have a statistician look at the study, see if things are, if your recruitment numbers are as you're expecting, if the primary outcomes, secondary outcomes still make sense, if the statistics still make sense, and you move on from there. Let's see, I have another question here. Just one second. To follow up on, this had to do with the question about the placebo for the Pompe disease. To follow up on the earlier question, in one of your earlier slides you mentioned that placebo does not necessarily mean inactive product. That is correct. In the Pompe community with a commercial product available, could the commercial product be considered a placebo? With a commercial product available, could the commercial product be considered a placebo? Now, you could consider, so I understand there are two products to treat the alpha-glucosidase deficiency. So, in other words, would you consider that one arm would be the active comparator and then you would be using the active comparator, let's see, as compared to the experimental new product? Yes, so certainly, I mean, you could be doing, in other words, your question would be designing a superiority study. So is a new product any better than the old product? Is it different from the old product? Absolutely, that would be a completely reasonable type of trial, absolutely. Yes. So in other words, normal treatment versus product X versus normal treatment versus product Y and so on. Absolutely. Yes. And that would make sense. If you have a product that's already approved for treatment, then it seems that it would be most reasonable to use what's already approved. Now, this does get a little complicated because the manufacturers in the U.S. are not mandated to show superiority, inferiority, and so on. What they're there for is to show that the drug works. So typically, if a pharmaceutical company is designing a product, then they probably would want to show the difference from placebo. I mean, they don't want to show that their product isn't any better than, whatever is already out there. So that does get a little complicated. But the point there is that if there's already a product labeled, then it probably would be reasonable to compare that labeled product against the new product. But again, this does get complicated about who's footing the bill for this. Let's see. So another way to look at adaptive clinical trial designs is a really nice project called the I-SPY 2 trial. And this is a project where they wanted to use a multi-arm trial. And so instead of having A versus B, what they had was patients being recruited, and then there was a multi-arm randomization. So, and just to read to the bottom of this, as with the Herceptin model, patients are being matched with experimental medicines that are designed to target a particular molecular subtype of breast cancer. But unlike in other trials, I-SPY 2 investigators are testing up to a dozen drugs from multiple companies, phasing out those that don't appear to be working and subbing in others without stopping the study. And this is a really nice, and I think this is probably going to become a more popular way of testing drugs because you set up the infrastructure, which is very complicated, which I'll get to in a minute. So why not have multiple arms? Why not compare? You don't have to do placebo against X or X against Y, but why not X against Y against A against B against C? And then as patients, don't do well on arm 3, then you can randomize them to, or they move to another arm. So I think that's a really nice way of setting up the infrastructure for these, which I think is sort of the coming trend here. Another term, superiority versus non-inferiority. This is a subtle point, but it does come up fairly frequently. A superiority trial means that one arm is better than the comparator, tends to need less patients to statistically prove this. A non-inferiority trial means that one arm is not worse than the comparator and tends to need more patients. Getting back to the point I made before, how many patients do you need for a clinical trial? And again, the point of the adaptive clinical trial designs is to reduce that number. And what you're doing when you set up a clinical trial is to make some assumptions about the sample that you're studying versus the entire population of patients with the disease. More patients means higher trial costs. That's just a fact of the matter. It means better statistical power, but sometimes it's just simply not possible to recruit that number of patients, and then you have to go back to the FDA and talk to them about what you should do. Timeliness of trial, sometimes becomes problematic with being able to perform a clinical trial. I was at FDA as a clinical pharmacology reviewer during the time when Gleevec was approved for chronic myelogenous leukemia. And the issue there was that there were changes in practice now that Gleevec or imatinib was approved or was at least available, that it became more and more difficult to randomize patients to interferon, which tend to have a lot more side effects than the imatinib did. We've had the experience testing lorazepam against diazepam for children in status epilepticus. However, right smack in the middle of the trial, the emergency transport personnel started administering that class of drugs, the benzodiazepines, in the ambulance, and so that took out a lot of patients that we could have normally enrolled because they were getting pretreated. Stopping a trial can be stopped during the interim analysis, meaning an analysis sometime in the middle of the trial. There are typically stopping rules that are embedded in a trial where the trial can be stopped for futility, meaning that you can enroll every single patient on planet Earth and never come up with an answer to the question. It can be stopped for efficacy, that it's completely clear that the drug you're testing is better than whatever the comparator is, and it's unethical to continue enrolling patients onto the test, to the test agent, or the non-test agent, because it's so clear that the test article is so much better than what they're being randomized to. It can also be stopped for safety if there's a safety signal that it's clear that patients are having serious adverse events or what have you from the test arm, then the trial needs to be stopped. For clinical trials, for the design, what you need to include is what is your specific question, what are your efficacy and safety endpoints that would be clinically meaningful, is your endpoint or outcome measure validated? This is a big problem. Sometimes, especially for children, the outcome measures have not been validated yet, and so it's not clear whether the outcome happened because, or it's just not clear how useful the outcome measure is. Can this study be done? Is it feasible? Can you afford it? Can anybody afford it? These are all questions because a lot of times studies aren't done for these reasons. For performance, who will manage the trial? Are there enough accessible patients who will meet your enrollment criteria? Sometimes when people develop trials, the inclusion-exclusion criteria are very stringent, and so you wind up excluding pretty much everybody who would possibly be enrolled. For example, if you're looking at a trial of pain and every child is taken out because they can't get Tylenol or ibuprofen, I mean, they'll pretty much eliminate, the patients that would be available. And I'm a pediatrician and a pharmacist, so this is my issue, is that do you have a dosage form that your patients can take? And this is frequently a problem in children. Okay, for developing a new drug, can be a new, for example, a new molecular entity. This is not a Me Too drug that looks like other drugs. Typically there have to be meetings with the Food and Drug Administration under the pre-IND portion. That's where the preclinical work is done, the preclinical toxicology, including reproductive toxicology. Typically 3 models are used, 2 rodent and 1 non-rodent. Again, the subtext here is there's an assumption that this toxicity has anything to do with humans, and sometimes it does, and sometimes it doesn't. The preclinical work is also done to look at efficacy, the estimate of a first-in-man dose, and some dose escalation plans. There is also a meeting with FDA at the end of Phase 2 to discuss the results so far and plans for a Phase 3 trial. Another term that's frequently used is the power. The power to detect an effect if there isn't one is a false positive, and that's considered a type 1 error. And the probability of making a type 1 error is alpha. The probability of making a type 2 error, which is called beta, is the situation where you do not detect an effect if there is one or a false negative, and this is a type 2 error. And if the trial is these are issues for statisticians. But if the trial is underpowered, what the assumption is that there is a high probability of making both a type 1 and a type 2 error. And the problem there is that you may, again, have incidental findings where, again, it looks like there's an effect when there isn't one, or it looks like there is not an effect when there actually is one. So these are statistical terms. The statistician typically determines the study power and performs the interim analysis. And the reason why those observational studies are so important is because it gives the statistician an estimate of what they assume will be the number of patients necessary to see a 10% improvement in the 6-minute walk test, or a 20%, whatever your company and the FDA agree should be a clinically relevant meaning. And again, the statistician determines the study power, determines the number of patients that are required to show that power, and performs the interim analysis. Now, the reasons for failure of clinical trials, and unfortunately this happens too many times, not all the time, but it does happen fairly frequently that clinical trials fail. Point number one is the preclinical animal model did not correlate with any human outcome, including efficacy and safety. So in other words, the drug was too toxic, the drug wasn't absorbed, the drug didn't work. The wrong dose was used. This is a problem. So there needs to be good dose escalation to make sure that you're giving patients the right dose of the drug. So it may be that the drug might work, but you gave half the dose or a third of the dose. You were unable to recruit eligible patients. This is a problem. There's patient dropout, loss to follow-up, and some of this may be that the study is too complicated, that the follow-up was too long, and no one could be, as in that previous question, traveling 700 miles to get somewhere. Could be that the biomarkers weren't validated or not correlated with the outcomes, that there were unvalidated outcome measures used, such as behavioral outcomes that may have not been validated. There could be failure to adhere to good manufacturing practices. The little c is current, so current good manufacturing practices, current good laboratory practices, current good clinical practices. And if this is the case, when the FDA reviews the data and audits the sites, they will see that they were not in compliance, and so therefore it puts the study results at risk. There may also be a discrepancy between the sample of the smaller number of patients in the phase 2 trial and the population in phase 3. So things may look really good in phase 2, but then it bombs in phase 3, and that's a problem. Okay, and again, those practice guidelines, GMP and so on. So these are the clinical trial elements. I just wanted to buzz through this. So we have the Food and Drug Administration and the European Medicines Agency. We have the sponsor. We do clinical trials at the NIH in my branch, so in which case we're the sponsor, but typically this would be the pharmaceutical industry. We also have the Data Monitoring Committee down there on the left side, which is an typically unblinded, non-conflicted group of experts in different areas. And for my purposes, we also have a Data Coordinating Center, which is separate from the sponsor. We have a contracting officer who negotiates the contract between the sponsor and the recruiting sites. There's a principal investigator that's responsible for the conduct of the trial, a nurse coordinator who's responsible for patient recruitment, which is an invaluable part of this piece. We have an institutional review board at the recruiting sites, an investigational pharmacy, and then obviously the most important people, the patients and the parents. So in terms of who's who here, the sponsor pays for the trial, develops the protocol, ultimately responsible for study conduct. The principal investigator is responsible for performance of the trial as written in the protocol. The Data Coordinating Center, and again this has to do probably more with NIH, although there are data coordinating centers for pharma, is usually a contract research organization or a CRO, which monitors the sites, tracks enrollment, takes care of drug accountability, we shipped you the drug. Where is it? Who did it get dispensed to? Was it, is it accounted for at the end of the study? And performs regulatory activities such as submitting investigational new drug applications to the Food and Drug Administration or to the EMA. So in terms of trial performance, we have trial management, and then tracking recruitment at the sites. I don't think we need to belabor this. The Data Monitoring Committee, also known as the Data and Safety Monitoring Board, DSMB, so take your pick on nomenclature there, is an external, unconflicted committee. Consists of a chair, a statistician, an ethicist, a patient representative, and a specialist in the area of interest of the clinical trial. So, for example, Pompe disease, we may be talking a geneticist, neurologist, what have you, cardiologist, pulmonologist, and so on. Let's see, I think that this has more to do with NIH than probably with you. Just to point out that the research pharmacy controls the study drug supply, dispensing, disposal, and so on. And then we have the laboratory staff, and then we also have the pharmacologists, and they're invaluable here because they set the study design, the dose selection, and determine the concentration-effect relationships. Now, I wanted to tell you a little bit about a study that we did, and I'd mentioned this in passing before, and this was about our clinical trial comparing lorazepam and diazepam for status epilepticus. So FDA had this question, and the question was, is lorazepam better than diazepam in treating status epilepticus in children? Is lorazepam safer than diazepam? Is there a difference in efficacy and safety in children of different ages? Now, the definitions became a little touchy because we had a lot of discussions with FDA about the definition of status epilepticus. Was it seizure activity for 5 minutes, for 10 minutes? How long would the seizure be considered to be status epilepticus, not just the self-limited seizure? And then what was the definition of seizure recurrence? And was the drug safer in terms of the patient needing assisted ventilation, such as a bag, valve, mask, oxygen, or intubation? Eligible children. We had a lot of questions about this. So how many children in the U.S. seen in the emergency room are in status epilepticus? And we went through the ICD-9 codes, which are the billing codes, insurance databases, but the problem with those numbers is that they really relate to billing, not necessarily to diagnosis. Although they should be the same, sometimes they're not. And then in order to determine which sites should be included, the question was to the sites, how many children with status epilepticus were seen in the emergency rooms you are planning on contracting with. The study endpoints included a consensus on endpoints for the efficacy endpoints and the safety endpoints. The question really was feasibility. Can this study be done? The consent process, as you can imagine, for a child in status epilepticus is a very critical, life-threatening situation here. Consent would be very difficult in an emergency room. Were there a number of eligible subjects necessary that could be pre-consented, for example, in the in the neurologist's office who were known to have seizure disorders, or were the inclusion-exclusion criteria too restrictive? So these were a lot of questions that we had. So at the end of the day, the FDA issued us an exception from informed consent. This is the first pediatric exception from informed consent. In the Code of Federal Regulations, there's a total outline of this process, including the exception from informed consent process, including community consultation, a lot of questions about who the community was it a geographic community, was it the community of children with pre-existing seizure disorders, and there was a large learning curve for the institutional review boards to familiarize themselves with the new process. Now, in the middle of the study, this was when the emergency personnel started administering anticonvulsants in the field. perfectly reasonable, but this was a new process, and so we ended up having to recruit a lot more sites because some sites were not able to recruit because their area started administering anticonvulsants en route. So I guess my summary on this is that the clinical trials are extremely complex. They involve a lot of people. They're very expensive. They must be well planned and executed. They must be performed in compliance with FDA guidance, and this is critical for my branch because we're submitting these data to FDA for labeling changes. So this is not an academic exercise. This is actually a regulatory exercise. And that the trial design and the endpoint should be discussed with the FDA early in the process. I think that's all I wanted to say. I just wanted to let you know that if you would like to contact me at any point, there's my name, my email address through the government, and my phone number. So don't hesitate to call or email. I get email and phone calls all the time. This is what I do. So please, feel free to call. I won't necessarily know the answer to your question, but I'll be glad to find out. Thank you.
Thank you, Dr. Zajicek. Participants can submit a question by typing their question in the rectangular input field below the chat window. Just type your message and press Enter to send it. As a reminder, a PDF of the presentation slides is available for download using the Program Materials link in your confirmation and reminder emails. At this time, no further questions have been submitted.
Okay. And again, I appreciate your questions as they were. And again, if anything comes to you later on that you hadn't thought of, please feel free to call or email me. I'll be happy to talk to you or email you back. Thank you.
Thank you, Dr. Zajicek. This concludes today's AMDA webinar. Thank you all for attending.
Thank you very much. Bye-bye.